molecular biology topics, it can tend to be a little distant from real living creatures. When we started talking about eDNA, we were seeing this connection to a greater ecological perspective on biology.
eDNA is environmental DNA, right? So we look for organisms without ever seeing them.
So sometimes it's like slowing things down and trying to figure out what's going on, and that's when you start connecting the dots. The science begins when the procedure ends.
So one of the things I like to do with my students is teach them about the use of journals in academic research. And the one I stumbled upon was Hellbenders and eDNA. And when I started reading about it, we had just started this idea of a genetics lab here at Seneca, and I was working with Mr. Lavelle on some things we could do up in the lab. And I took the journal article up to him and said, look at this, it's really cool. And he read it and responded back on the computer, I think we can do this in lab.
The Center for Biotechnology Research is focused upon offering students authentic research experiences in a wide variety of areas, including genetics, microbiology, as well as genetic editing, and we're going into a few other areas too, like fluorescent microscopy and biofilm research as well.
It is totally self-motivated, so it's not a class, it's not on the schedule anywhere. There's not a credit associated with it. It's totally students that want to learn about research.
What we're seeing over the few years that we've been doing this is this gradual development of a student into what we see as a really capable scientific researcher.
My name is Sydney. I am a Gene Tech 4.0 and I'm in grade 12, or I'm a senior. Our research project, I'm working with another Gene Tech 4.0, and we are analyzing eDNA and looking at the presence of hellbenders, or lack thereof, in the streams.
My name is Courtney Beals. I'm a senior this year, and I'm a Gene Tech 4.0. This year I'm looking at the Eastern Hellbender and whether it's present in the big Connoquenessing Creek and the little Connoquenessing Creek right around our school and in the surrounding Harmony area. We just want to try to pinpoint where these hellbenders could be located and understand their habitats a little more.
We have many streams in Pennsylvania that can support hellbenders. They don't always support hellbenders because of past practices and environmental damage.
My name is Maggie Monahan. I'm in 11th grade and I am a Gene Tech 2.0.
My research question was trying to find where hellbenders are in the little Connoquenessing Creek around the school, and so I tested 5 different locations, starting from the bridge coming onto campus to the tennis courts at the far edge of campus, and compared the trials from that to see where the hellbenders most likely are.
My name is Perry Gagas. I'm in 11th grade and I am a Gene Tech 2.0. My research question this year, I was looking to see if there was a correlation between water properties of streams that housed hellbenders and streams that did not house hellbenders. So I tested dissolved oxygen, pH, and conductivity of those streams, and then obviously used our DNA purification process and compared the results between those two to see if there were differences in those levels.
So mostly what we do, and there's other ways you can do this, but mostly what we do here is we pull water samples, we filter and purify the DNA of anything that is in the waterway that we're looking at. And then we have primers specific for what organism we're looking for. And so we run those in our qPCR machine.
PCR machine is basically, generally, a gene copying machine. So you don't copy the DNA from an entire organism, you just copy a specific gene. And in our case, we copy a gene that is very unique to the species we're looking for. And the one we usually look for is the hellbender.
If we get a hit, we would get a positive result, and then we could say, although, we're not entirely certain, right? So we could say that we think we have presence of that particular organism in our waterway that we're looking at.
I really think it's super interesting to see that there's a way where we can detect if a species that's endangered or threatened lives in our surrounding areas without even having to see it.
It allows researchers to track and study potentially vulnerable organisms or even endangered organisms without having to invade their habitat.
So I think it's really good in conservation efforts. I mean, I know, just relating it back to hellbenders, not that they're endangered, but the population's on the decline because they're such sensitive organisms. So I think being able to, again, test for them, see if they're present in a certain waterway without disturbing them and potentially harming the population even more is really, really big benefit.
You can learn a lot from it without kind of that interference.
Well, the results were a little inconclusive, but one of my streams, Brush Creek specifically, I had very good results on, and it really, based on my research of hellbenders, it really fit like the criteria. It had a more neutral pH, lower conductivity levels, and higher dissolved oxygen. But the other two streams, which were Little Swickley and Big Swickley, kind of flipped on me a little bit. Little Swickley, you think that it would have the better water quality, and it did, but it did not. I did not have a hit on hellbenders, but Big Swickley, I did have a hit on hellbenders.
I found that the area by the softball fields has the strongest hits consistently, and so we believe that the population is near there.
Our results have been successful this year because we discovered that there is a likely presence of hellbenders in the Little Connoquenessing Creek, as well as the convergence of the Little and Big Connoquenessing Creeks. And we determined this by taking our own samples of those water bodies.
To see over the course of a year, or even we've had some students that I've been working with for now 3 years, to see how they have grown in their lab technique and ability is just amazing. So we have some students that I would stack up against any university level researcher for sure as far as their lab technique. The other thing that we see a lot that's really encouraging and really rewarding is the students want to come to lab. We have students signing up all the time. They're ready for the next step. They're ready for the next experience. So to see how they become excited, they want to be in the lab. That's really cool. And so that's really kind of heartwarming, and it just makes us feel like, that we're doing something really beneficial. It's all for the benefit of the student. It's to get them, what they want to do now, but also it's preparing them for the future, and even in a lot of cases steering them towards what they want to do in the future. Very rewarding.
Before doing the genetics lab, I knew I loved science, but I didn't really know what the research component of that would be like. And so just getting to be able to do this has really, I really love doing this, and just kind of figuring that out was really cool.
Next year I plan to continue my research either by doing a similar experiment, like testing for a correlation, like between, maybe a different property of streams, and if they have hellbenders or not, or developing a primer could be another route. I know a lot of seniors have done that. In terms of after high school, I mean, of course I want to do research in college, especially if I'm going to the medical field. That's very beneficial. So
I plan to continue my scientific research in college. I'm going to a larger university, so I hope to kind of have my foot in the door with the skills I've already built from this genetics lab, and hopefully work in a larger scale biology, certainly based lab of a professor, or just to continue my love of research.
I plan on going to a school to study something STEM related. Right now I'm looking into pharmacy or pharmaceutical research, and this lab has really inspired me to go further on with research. It has inspired this discovery within me. I want to continue with research in whatever way that may be, because I really love to almost contribute to the unknown, figure that out instead of always just being in a classroom and having things being told to you. In this case, you're figuring things out to help the greater scientific world.